First meiosis, prophase first, metaphase first, anaphase first, and telophase first. Then second one is meiosis second. Meiosis second is prophase second, metaphase second, anaphase second, and telophase seconds. These are the two types of reductional division of meiosis. So we have prepared some PPT. We see the PPT of meiosis cell division. Okay, meiosis is a eukaryotic cells, plant, animals, and fungi reproduce sexually. It is a process of chromosomal reductions, which means that a diploid cell is reduced to form haploid cells. The haploid cell produced by meiosis or germ cell, also known as gamete, sex cells, or spores in plant and fungi. These are essential for sexual reproduction. Two germ cells combine to form a diploid zygote, which grow to form another functional adult of the some species. The process of chromosomal reduction is important in the conservation of chromosomal numbers of the species. Meiosis occurs in two distinct phases. First one is meiosis first and second one is meiosis second. First, meiosis first is first phase is prophase first. Prophase first again divides five phases. That is five subphases, means which first one is liptotin, second zygotine, third one pachytine, fourth one diplotins, and fifth one is diakinesis. First, liptotin. Liptotin is also known as liptonema. Liptotin is also known as liptonema. The chromatin condense of chromatin condense to form visible chromosomes. Condensations and coiling of chromosomes occurs long thread-like shape, and they develop bed-like structures called chromosomes, and the chromosomes in the nucleus appears like a bucket in animal cells. This stage is also called as the bucket stage. Then second stage is zygonema. Zygonema is also known as zygotine. Okay, zygotine. Zygotine stage being with the pairing of homologous chromosomes. Pairing of homologous chromosomes, and it will be occurs just synopsis. It is called as synopsis. The paired homologous chromosomes are connected by a protein containing framework called as synaptimal complex. ज्या ठिकाणी प्रोटीन कंटेनिंग जे फ्रेमवर्क्स निर्माण होतो त्या फ्रेमवर्क्सला सिनॅप्टिमल कॉम्प्लेक्स म्हणलं जातं. The synaptimal complex helps to the stabilize a pairing of homologous chromosomes and facilitate the recombinations or crossing over. एकमेकावरती ओव्हरलॅप होत असतात, क्रॉसिंग ओव्हर. The synapses might be at one or more points along the length of the homologous chromosomes. Then synopsis happens when the homologous pairs join the synaptimal complex form. ज्या वेळेस homologous pairs क्रोमोझोम जे पेयर्स जॉईन होतात त्या जॉईनच्या ठिकाणाला काय म्हणतात? सिनॅप्टिमल कॉम्प्लेक्स म्हणतात. नेक्स्ट, Pachytene also known as pachynyma. Pachynyma is the third main event of the prophase. First occurs non-sister chromatids of homologous chromosomes. Pair exchange part of or segment is chiasmata forms where these exchanges have occurred. Each chromosome is now different to its parents' chromosomes but contain its some amount of genetic material. The process of mutual exchange of chromatin materials between one non-sister chromatids of each homologous chromosome is known as crossing over. Crossing over. हे बघा some figures of prophase first. See here, first one is a liptotins. Liptotins is a nuclear membrane. Nuclear membranes formed through the condensation of chromosomes. Condensation of chromosomes. Second one is zygotin. Zygotin is also known as zygonyma. It's formation of bivalents. Bivalents of homologous chromosome. Formation of synaptimal complex and synthesis between homologous chromosomes. Here, synthesis between homologous chromosomes. Third one is the pachytins. Pachytin stage is also called as pachynyma, and here occurs the crossing over. In the pachytins, pairs homologous chromosome or homologous pairs in the occurs a crossing over. Then fourth one is the diplotins. Here occurs crossing over and to form a chiasma. Here the chiasma, red colors and blue colors overlapping the chromosomes. They will be called as chiasma and dissolution of the synaptimal complex. Here, see the synaptimal complex. And last one is dikinesis, dissolutions of nuclear membranes. This is the prophase first. It will be again divided 5 stages. That is first one liptotin, zygotin, pachytin, diplotin and dikinesis. Diplotins. The synaptimal complex dissolve and chromosomes pairs bear to begin to the separate and synaptimal complex appear to the dissolve, leaving chromatids of the paired homologous chromosomes physically joined at one or more localized points called as chiasma. In diplotins, chiasmata move towards the end of chromosomes in a jeep-like manner. Dikinesis is marked by the termination of chiasmata and assembles of the meiotic spindle to separate the homologous chromosomes, and the nucleolus disappears and nuclear envelopes break down. This is the first prophase. Again divide 5 stages. Then second one is the metaphase first. Metaphase first is a bivalents aligned at the equatorial plate and microtubules from the opposite pole attached to the pair of homologous chromosomes. Then second one is anaphase first. The two chromosomes of each bivalent separate and move to the opposite end of the cells. The sister chromatids are attached to each other. And fourth one is telophase first. The nuclear membrane reappears and is followed by cytokinesis. This gives rise to the dyad cells. Dyad cell. This is the meiosis second phase. First one is prophase second, second metaphase second, prophase second, metaphase second, and metaphase seconds, and third one anaphase seconds, and fourth one is telophase seconds and cytokinesis. Here diad cells. Here the diploid daughter cells forms and sister chromatids is separated. ह्या ठिकाणी हे दोन आणि हे दोन काय होतात चार टेट्रॅड्स न्यूक्लियस आर सेपरेटेड म्हणजे फॉर्म होतात. Meiosis seconds. First one is prophase seconds. The chromatin condenses to form visible chromosomes again. The nuclear envelope and nucleolus disintegrate and spindle fibers begin to appear. No crossing overs occurs. या ठिकाणी प्रोफेज सेकंड मध्ये क्रॉसिंग ओव्हर होत नाही. दूसरा है मेटाफेज सेकंड. The chromosomes are connected to the centroid poles at the kinetochores of sister chromatids through the microtubules. They also get aligned at the equator to form the metaphase plate. Third one is anaphase second. In this phase of meiosis second, there is a simultaneous splitting of the centromeres of each chromosome, and the sister chromatids are pulled away toward the opposite poles as the chromatids move toward the pole. Towards म्हणजे separate होणे, and the kinetochores is the leading edge with the chromosomal arm trailing. Next है telophase. The telophase, this is the fourth phase of the meiosis seconds. This is meiosis seconds, telophase seconds. The chromosomes dissolve again into an undifferentiated lump and nuclear envelope develops around it, followed by cytokinesis. Telophase seconds make the end of meiosis and to form 4 haploid daughter cells are formed as a result. ह्याच्यामध्ये 4 haploid daughter cells निर्माण होतात. कोणते न्यूक्लियस daughter cells are formed. Significance of meiosis. Importance of meiosis म्हटलं तरी चालेल. Meiosis is a responsible for the formation of sex cells or gametes that are responsible for sexual reproduction. त्याच्यामध्ये या meiosis मध्ये responsible काय आहे? Sex cells आणि gametes. These are the two organs are responsible for the sexual reproduction. And it activates the genetic information for the development of sex and deactivate the chorophytic information. It maintains the constant number of chromosomes by halving the chains. This is important because of chromosome number doubles and after fertilization. In this process, independent assignments of materials and maternal and paternal chromosomes takes place. Thus, the chromosomes and the traits controlled by them are restful. The genetic mutation occurred due to the irregularities in the cell divisions by meiosis. The mutations that are beneficial are carried on by natural selection. And most and important is Crossing over produce a new combinations of trait and variations. Trait and variations. Thank you. Uh, हे बघा